Hello everyone, we are here to present a topic of physiology of vision, pupil and light reflex. It's an important topic for ophthalmology for part 2. My name is Bhaskar Chandra, third pro part 1 student of RGKMCH Medical College. So now we come to the lecture of physiology of the vision. So, if what is vision? The light passes through the cornea and pupil at the front in the front of the eyes. Suppose here is the object and this is the eyeball. The light passes through the cornea and the pupil at the front of the eye. It is focused by the lens into the retina. There is the retina and the cornea and the lens converge the light so it passes through the vitreous gel. Here lies the vitreous gel in the posterior segment of the eye and falls on the retina. The retina converts light to the electrical impulses and passes by the optic nerve to the visual cortex. So if we consider an eye is an optical instrument. It is similar to that of the camera of the old age. Eyelids act as the shutter of the camera. Cornea, crystalline lenses, along with the aqueous humors, act as the focusing systems of the lens of the camera. Combined in the actions from a very strong refracting systems of the short focal length. Iris acts as the diaphragm. This is the iris. Iris lies in the anterior portion of the lens and acts as the diaphragm which regulates the size of the aperture, that is pupil. You know the exterior eye have blue iris and a majority of the Indians are of black and brown iris. Yes, you should know. And the amount of the light enters the eyes. Choroid helps in forming the darkened interior of the camera. The retina acts as the light sensitive plate or film on which the image is formed. We now discuss about a clinical feature, suppose the clinical disorder, that is reduced time. So you can see this is the classical picture of reduced eye taken from a prestigious book. So I have some points about this reduced eye. Optics of the normal eye is very complicated. Therefore, for better understanding, listing simplified and the data in the imaginary eye called reduced eye. Simplified data of the listing reduced eyes are as follows. I have this brush and color. Okay, I choose this color. Okay. Simplified this I shall try to discuss it as a symbol. Reduced eye in principal point. This is the principal point. You can see what this is the P signs of थोड़ा सा साइज छोटा करता हूँ अच्छा principal point, this is the P and the lies at the 1.5 centimeter in front and behind of the anterior surface of the retina. P lies in front lies behind the anterior surface of the cornea. This is nodal point. In this is situated 7.2 millimeter behind the anterior surface of the cornea, and the anterior facial point, that is F, is 15.7 millimeter in front of the anterior surface of the cornea. Yes. So, and posterior focal point, that is F2. This F2, the previous was, this was F1, this is F2. F2 will be on the retina, and this is 24.40 millimeter behind the anterior surface of the cornea. Anterior focal point, that is F, small f, that is F is 17.2 millimeter, um, and will be 17.2 millimeter, that because there will be 15.2, sorry, uh, there will be 15.7 plus 1.5 equal to 17.2 millimeter. Okay, yes, I was right. Anterior focal length F is 17.2 millimeter. This is the anterior focal length, 17.2 millimeter, and posterior focal length F2 will be 22.90 millimeter. And the total dioptric and the power of the reduced eye will be plus 60 diopter, out of which about 40 plus 43 D is contributed by the cornea. This plus 43D is contributed by the cornea only, and plus 17 diopter of the crystalline lens. So this is about reduced eye. So now we will discuss about optical axis. Optical axis. This is the optical axis. Optical axis is the line passed through the center of the cornea P and the center of the lens N and meets the retina. And meets the retina on the nasal side of the fovea. This is the fovea, and visual axis of the line joining from the fixation point O and the nodal point of the fovea. This is the optical axis. Optical axis. This is the visual axis. So what will be the visual angles? We'll now come about visual angles. Visual angles, there are three types of the visual angles: angle alpha, angle gamma, gamma, and angle kappa. Angle alpha, this is an angle found between the optical axis and the visual axis wave at the nodal point. This is the optical axis and this is the visual axis. So this is the this will be the angle alpha. Angle gamma, between the optical axis and fixation axis OC at the center of the rotation of the eyeball. And angle kappa will be the axis of visual axis and the pupillary axis. The point P on the center of the cornea is considered equivalent to the center of the pupil. Practically only the angle kappa can be measured and is clinically significant. Only kappa. This is only the clinically significant. ये वाला कम का है. A positive angle kappa results in pseudo exotropia. This is pseudo exotropia, and the negative angle kappa is the pseudo esotropia. exotropia and esotropia. Pseudo, this is positive, and pseudo eso, this will be negative. So now we enter into the physiology of the vision. The main mechanism of involved in the physiology of the vision are initiation of the vision, that is phototransduction, a function of the photoreceptors rod and cones, processing and the transmission of the visual sensation, a function of the image processing cells of the retina, and visual perception, a function of the visual cortex and the related areas of the cerebral cortex. So here we want to see details about the we should keep in mind about that sense of the vision is a complex function of the two eyes. We have two eyes, no? Achha. And the central connections. The physiological activities involved in the normal functioning of the eyes, the maintenance of the clear ocular media, maintenance of the normal intraocular pressure, the image forming mechanisms, physiology of the vision, physiology of the pupil, physiology of the binocular vision, and physiology of the ocular motility. And the major factors responsible for the transparency, that is transparency, is the tear film, cornea, aqueous humor, and crystalline lens, and vitreous humor. So physiology of the vision is a we can see is a complex phenomenon which is still poorly understood. The main mechanisms are here we mentioned: the phototransduction, processing and transmission of visual sensations, and visual perceptions. So थोड़ा सा पॉइंट डिस्कस कर लेता हूँ. Initiation of the vision, that is phototransduction, is a function of the photoreceptors, that is rods and cones. Processing and transmission of the visual sensation is a function of the image processing of the cells of retina. And visual perception is a functional and visual cortex and related areas of the cerebral cortex. Let's go on the phototransduction. Phototransduction is about the rods and cones serves as a sensory nerve endings. It's for the visual sensations. The light falling on the retina causes the photochemical changes, cascade of biochemical reactions that results in generation of electrical changes. Photochemical changes occurring in the rods and cones are essential but similar. But the changes in rod pigment, rhodopsin or the visual purple, have been studied with more detail. The whole phenomenon is conversion of the light energy into the nerve impulse. It is also known as phototransduction. So what is phototransduction? The phenomenon of conversion of the light energy into the nerve impulse is known as phototransduction. Okay. So here we discuss the mechanism of the visual cycle. This is a visual cycle. Photochemical changes occurs here. So now we discuss this image. First, there will be the rhodopsin. First, there will be rhodopsin bleaching. Rhodopsin refers to the visual pigment present on the rods, the receptor for night scotopic vision. Its maximum absorption spectrum is around 500 nanometer. Rhodopsin consists of the colorless protein called opsin, coupled with a carotenoid called retinine. Retinine is the vitamin A aldehyde or 11-cis retinal. Light falling on the rods converts cis 11 retinal components of the rhodopsin into the 11 cis retinal through the various stages. All the transretinal is formed is so separated from the opsin. The process of the separation is called photodecomposition. जो प्रोसेस होता है 11 सीरीज रेटिनल से separation of the opsins is called photodecomposition, and this rhodopsin is said to be bleached by the actions of the light. There will be the light bleaches the 11 cis retinal, and there will be the rhodopsin regeneration. Rhodopsin regeneration. There will be the rhodopsin regeneration in the 11 cis retinal is regenerated from alt trans retinal separated from the opsin, and vitamin A then reunites with opsin in the rod outer segment to form the rhodopsin. This whole process is called the rhodopsin regeneration. Thus, the bleaching of the rhodopsin occurs under the influence of the light, whereas the regeneration process is independent of light, producing equally well in light and darkness. So, about we go to the visual cycle. What is visual cycle? Visual cycle. This whole case scenario is the visual cycle. In retina of the living animals under the constant light stimulations, a steady state must exist under which or rate at which the photochemicals are bleached is equal to the rate at which they are regenerated. The equilibrium between the photo decomposition and regeneration of the visual pigments is referred to as visual cycles. So now we go to the electrical changes. The activated rhodopsin. Activated rhodopsin will, following the exposure of the light, it triggers a cascade of the complex biochemical reactions. ये वाला reactions है. Light energy will convert rhodopsin to batho-rhodopsin, lumi-rhodopsin, then meta-rhodopsin 2, then again scotovision and all transretinal isomerase. That's this will be occurred. This is the cascade reactions. Then finally receptor activated rhodopsin following the exposure of the lights triggers a cascade of complex biochemical reactions which ultimately results in the generation of the receptor potential. This receptor potential will be regenerated in the photoreceptor cells, obviously in the retina. In this way, the light energy is converted into electrical energy, which is further processed and transmitted by the visual pathway. So, initiation and transmission of visual sensations. And the rods and cone cells serve as a sensory nerve endings for visual sensations. Stimuli for visual sensation divided into two types, that is inadequate stimuli and adequate stimuli. Inadequate stimuli produce glowing sensations called phosphene, and adequate stimuli formed by the visible portions of the light and light absorbed by the photosensitive pigments present in the rods and cone cells and photochemical changes occurs. Electrical impulses transmitted through the ganglion cells along the fibers of the optic nerves and others part of the visual pathway to the visual cortex. Visual perceptions. Visual perceptions. This is a sensation which results from the sensations of the retina with light. This is the four types: the light sense, the form sense, the contrast sense, the color sense. So we entered into quite deep into it. Um, the receptive fields, organizations of the retina and the visual cortex are used to encode this information about the visual image. The light sense. Light sense, it is the awareness of the light. The minimum brightness required to evoke a sensation is of light is called the light minimum. It should be measured when the eye is dark, adapted for at least 20 to 30 minutes. The human eye is an ordinary use throughout the day is capable of functioning normally over an excessively wide range of illuminations by a highly complex phenomenon termed as the visual adaptation. The process of the visual adaptation primarily involves dark adaptations, adjustment in dim illuminations, and light adaptations, adjustment to bright illumination. So now we, in light sense, this obviously refers to the appreciation of the light for the awareness of the light, and it is measured when the eye is dark adapted for at least 20 to 30 minutes. We have already discussed, and the red cells are more sensitive than cone in low illuminations. Then it is called scotopic visions. Cone cells are more sensitive in the bright light called photopic visions. So here is an important point that is dark adaptations. डार्क एडेप्टेशन ये शॉर्ट नोट आता है और डार्क एडेप्टेशंस अबाउट it is the ability of the eye to adapt itself to decreasing illumination. When one goes from the bright sunshine into the dimly lit room, and so one cannot perceive the object in the room until sometimes has elapsed. During this period, eye is adapting to the low illumination. The time taken to see in the dim light illumination is called dark adaptation time. The rods are much more sensitive to the low illumination rod. Rods are for scotopic, for night visions, and are for cones are for day vision or color vision. So there lies a dark adaptation curve. There it plotted with the illumination of the test objects in the vertical axis and the duration of the dark adaptations along the horizontal axis shows that the visual threshold falls progressively in the darkened rooms for about an hour until the relative contrast value is reached. Further, the dark adaptation curves consist of the two parts. Here we This is for cone adaptation, and this is for rod adaptations. This is the stimulus intensity, and this is the time. So when fully dark adapted, the retina is about 1 lakh times more sensitive to the light than when it is bleached. And delayed dark adaptations about in diseases of the rods, example retinitis pigmentosa and vitamin A deficiency. Another is light adaptations. Light adaptation: when one passes suddenly from a dim to a brightly lighted environment, it is opposite to the dark adaptation. The dim light to light the environment, the light seems intensely and even uncomfortably bright until the eyes adapted to the increased illuminations and the visual threshold rises. The process by means of which retina adapts itself to the bright light is called light adaptation. Unlike dark adaptation, the process of the light adaptation is very quick and occurs over a period of only 5 minutes. Strictly speaking, light adaptation is merely the disappearance of the dark adaptations. So from the scene, it is ability to discriminate between the shapes of the objects. From the discriminate between the shapes of the objects, cones plays the major role. Therefore, from sense, it is more accurate in fovea. Visual acuity by Snellin's chart is measured of from sense. So the clinical test determining the visual acuity measures from the sense of reading ability of the eye. Thus, broadly, resolution refers to the ability to identify the spatial characters of a test figures. The test targets in this test may either consist of letters, Snellen charts, or broken circles like Landor's ring. More complex targets including gratings and checkerboards patterns. There are two important words, that is recognition and minimum discrimination. Recognition, it is that faculty of virtue of which an individual not only discriminates the spatial characteristics of the test pattern but also identifies the pattern with which he had some experience. Recognition is thus a task involving cognitive components in addition to the spatial resolution. For recognition, the individual should be familiar with the set of the test figures employed in addition to being able to resolve them. Two more common example recognition phenomenon is identification of faces. The average adult can recognize thousands of faces. Thus, from sense is not purely the retinal function, as the prescription of its composite from example letters is largely physio-psychological. Minimum discriminable refers to the spatial distinction by an observer when the threshold is much lower than the ordinary acuity. The best example of the minimum discriminable is vernier acuity. Vernier acuity, which refers to the ability of determine whether or not two parallel and straight lines are aligned in the frontal plane. Sense of contrast. It is the ability of the eye to perceive slight changes in the illuminance between regions which are not separated by the definite borders. Loss of the contrast sensitivity results in mild foggings of the vision. Contrast sensitivity is affected by the various factors like ages, refractive errors, glaucoma, and am by sorry um, amblyopia, diabetes, optic nerve disorder, and lenticular changes. Further, contrast sensitivity may be impaired even in the presence of the normal visual acuity. So, what are the how will you test the contrast of the sense? In clinical practice, the contrast sensitivity can be measured by using any of the following charts with letters and strips represented in the various gray. One of the important is Arden's gratings. Another are the Cambridge low contrast grading and Bailey Robson's contrast sensitivity chart. The contrast sensitivity decreases from one triple to the next. Visual charts and functional acuity contrast test. Next is color vision or color sense, the sense of the color. It is the ability of the eye to discriminate between the different colors excited by light of the different wavelengths. Color vision is a function of the cones, better appreciation in the photopic vision than in the dim light or scotopic vision. Theory of the color vision. This is the important theory of the color vision is trichromatic theory. The trichromacy of the color vision was initially suggested by Young and subsequently modified by the uh, uh, Helmholtz. Hence, it is called the Young-Helmholtz theory. It postulates the existence of three kinds of the cones, each containing a different photopigments, which is maximally sensitive to one of the three primary colors, red, green, and blue. The sensation of any given color is determined by the relative frequency of the impulse from each of the three cone systems. So, in trichromatic theory, there is the red sensitive cone pigment, there is a green sensitive cone pigment, and blue sensitive cone pigment. The red sensitive cone segment, what the red sensitive cone pigment, this is also known as erythrolabel or long wave length sensitive cone pigment. Red is also known as LWS, that is erythrolab or long wavelength sensitive cone pigment. Absorbs maximally in the yellow portions with a peak of 565 nanometer. But its spectrum extends far enough into the long wavelengths and sense red. Then we come to the green. Green sensitive cone pigment. Green sensitive cone pigment is MWS or medium wavelength sensitive cone pigment, or sometimes called some books chlorolab. Absorbs maximally in the green portions at the peak of 535 nanometer. Then we comes to the blue. Blue sensitive cone pigment or cyanoLab, that is short wavelength sensitive cone pigment, SWS. This absorbs maximally in the blue violet portions of the spectrum at the peak of 440 nanometer. Then we come to the theory of the color vision. This is opponent color theory of hearing. The opponent color theory of hearing points out that some colors appear to be mutually exclusive. There is no such color as red screen. Such phenomenon can be difficult to explain the basis of the trichromatic theory alone. In fact, it seems that both theories are useful. Color vision is trichromatic at the level of the photoreceptors. Color opponency occurs at the ganglion cell onwards. Color blindness. In color blindness, mechanism to appreciate one or more primary colors is either defective, anomalous, or absent anopia. It may be congenital or acquired. Congenital color blindness. It is a hereditary condition affecting males more than females. Males are affected 3 to 4% and females rarely 0.4%. It may be of the following types. Dyschromatopsia. Dyschromatopsia is only 2 or 3 primary colors can be distinguished, and achromatopsia is the partial or total absence of the color vision, only able to perceive black, white, and gray shades. Acquired color blindness. This may follow the damage to the macula or optic nerve. Usually, it is associated with central scotoma or decreased visual acuity. Blue-yellow impairment is seen in the retinal lesions such as CSR, macular edema, or shallow retinal detachment. Red-green deficiency is seen in the optic nerve lesions. Optic neuritis, levers optic atrophy, compressions of the optic nerve. Acquired blue color defects is the blue blindness or occurred in the old age due to the increased sclerosis of the crystalline lens. It is owing to the physical absorption of the blue rays by the increased amber color pigment in the nucleus. Test for the color vision. The pseudo-isochromatic chart vision, that is Ishihara tests, Ishihara plates, Edridge green lantern test, Parkinson's Orth Munsell's 100 U test, Paris Orth D15 hue discrimination test, City University color vision test, Nichols anomaliscope and homologous Wolf's test. Pupillary reflexes. Light reflects when light is shown in one eye. Both the pupils constricts. Constrictions of the pupil to which light is shown is called the direct light reflex, and that of the other pupil is called the consensual or indirect light reflex. The light reflex is initiated by the rods and cones. Pathway of the light reflex includes afferent fibers extend from the retina to the pretrial nucleus in the midbrain. This travels along the optic nerves to the optic chiasma when fibers formed in the nasal retina decussates and travels along the opposite optic tract to terminate in the contralateral pretectal nucleus. While the fibers from the temporal retina remains uncrossed and travel along with the optic tract of the same side to terminate in the ipsilateral pretectal nucleus. This is the pupillary light reflex. So, when we come to the right reflex, light reflex is internucleal fibers connect each pretectal nucleus with Edinger-Westphal nucleus. There is a Edinger-Westphal nucleus, and the connections form the basis of the consensual light reflex. Efferent pathway consists of the parasympathetic fibers which arises from the Edinger-Westphal nucleus in the midbrain and travels along the third oculomotor cranial nerve. The preganglionic fibers enter the inferior divisions of the third nerve and via the nerve to the inferior oblique which reach the ciliary ganglion to relay. The postganglionic fibers travels along the short ciliary nerve to innervates the sphincter pupillae and results in the constrictions of the pupil. Clinical significance of light reflex. Abnormal pupillary light reflex found in the optic nerve injury, oculomotor nerve injury, brainstem lesions, and depressant tracts such as barbiturates. Relative afferent pupillary defects is a condition in which people respond differently to light stimuli shown in one eye at the time due to unilateral or asymmetrical disease of retina or optic nerve. Only optic nerve disease occurs in front of the lateral cingulate body. Detected by swinging flashlight test, shines a pen light test towards one eye, then swing to the other, alternating weekly to observe the patient's pupil response to the light. Stimuli in case of the both pupils do not show a similar response to light stimuli shown in one eye at the time mainly dilate the pupil, then patient will diagnose with RAPD. If the conditions bilateral and the symmetrical bilateral APD helps early diagnosis to optic neuropathy and multiple sclerosis. So thank you.